Okay, so we are moving on to fluids and electrolytes. Our bodies are made up of anywhere between 45 and 75% fluid by weight. So you can see that's a pretty broad range. So it depends on a couple of different factors. So age is a big factor. Infants have the highest fluid percentage. They're usually upwards of like 75% fluid composition. And then the more you age, the more that decreases. So elderly people are going to be more towards 45%. And also, the relative amounts of adipose tissue or fat tissue that you have relative to your skeletal muscle, that also affects the percentage of your body composition that's made up of fluid. So adipose tissue is composed of about 20% water, and skeletal muscle is upwards of 75% water. So as a percentage of adipose tissue increases in your body, the fluid composition is going to go down. The fluid in our body is partitioned into intracellular fluid and extracellular fluid. So intracellular fluid, or ICF, that's going to be the fluid that's inside of our cells. About 2/3 of our total fluid is intracellular fluid located in our cells. So you can use these kind of pie charts to visualize this. So the barrier for that's going to be the plasma membrane. The plasma membrane is going to be what's keeping that intracellular fluid in the cells. Okay, so any fluid that's not inside the cells is going to be referred to as the extracellular fluid. The extracellular fluid is broken down into two different compartments. One of those is going to be the interstitial fluid. The interstitial fluid is the fluid that just kind of surrounds the cells. So I think we saw that in the last chapter when we were looking at reabsorption and secretion in the tubule system. All of those kind of zoomed in images you could see inside the tubule, and then the tubule epithelium, and then there was the interstitial area before you got to the capillary. So that's just kind of a fluid-filled area that kind of bathes our cells. So that's what interstitial fluid is. And then you've got blood plasma. So plasma is the fluid acellular, or the part that's not made up of cells, so the fluid portion of the blood. So interstitial fluid you're going to find surrounding the cells. Plasma you're going to find within the blood vessels. So the interstitial fluid makes up about 2/3 of the extracellular fluid. And then the blood plasma is going to make up the other third. The barrier separating the plasma from the interstitial fluid is going to be that wall of the capillary vessel. Okay, so our total fluid, we have 2/3 of it found inside of our cells. The last third is found outside of our cells. If you break down that extracellular fluid, 2/3 of that is interstitial fluid, which is surrounding or bathing our cells, and then 1/3 of that is found in the blood as blood plasma. The composition of intracellular fluid and extracellular fluid is different, and that's because they serve very different functions. So intracellular fluid, the fluid inside the cells, has a very high level of proteins and a high level of potassium. So that's because inside of the cell is where all of your protein synthesis is happening. So we're constantly making new proteins for a variety of different functions, and that's happening inside the cell. So we're going to have a lot of proteins in there. Also, we have a lot of potassium because we've got the sodium-potassium pump sitting on that plasma membrane of the cell, which is always pumping sodium out, but potassium in. So we're getting this constant influx of potassium. So we get a lot of potassium coming in the cell, so it's higher there. The extracellular fluid has a high level of sodium, calcium, chloride, and bicarbonate. So the interstitial fluid and the plasma, those are the two forms of extracellular fluid. Those have a very similar composition, but there's one main difference between them, and that's that the plasma has a lot of proteins while the interstitial fluid does not. And that's just because we have a need for proteins in the plasma. So we've looked at one of those in the past, which were transport proteins that we use to transport our lipophilic hormones through the blood because they don't like the aqueous environment of the blood. So we have a need for proteins in the plasma. There's not a huge need for proteins in the interstitial fluid because that's just the fluid that's just kind of surrounding and bathing our cells. So that's kind of the one main difference between interstitial fluid and plasma. Aside from that, their composition looks fairly similar, which looks different from the intracellular fluid. Fluid is always allowed to move between the different compartments. So in this case, we're talking about the compartments being a cell and the blood vessel with the interstitial fluid being in between those. So you can see that here. Here's inside the cell, here's the interstitial fluid, and here's inside the capillary. So these are all different compartments. They're bordered by different borders. So you've got the capillary wall separating the plasma from the interstitial fluid, and then you have the plasma membrane of the cell separating the interstitial fluid from the cell. So if you look at these as just different compartments, water is free to move from one compartment to the other based on changing needs. And basically those changing needs are going to be the relative osmolarity. So I like to think of osmolarity in terms of just concentration. I always just, in order to keep it simple in my head, I think of it like salt water. And we've got two containers of salt water that are kind of connected, and so the water can move wherever it wants, and the water is always going to go from the dilute area to the saltier area, right? Because it wants to kind of balance out. And so that's kind of how I try to think of this. So changes in osmolarity, so the water is always going to move. It's going to go through osmosis from the hypotonic area, which is the area that is the more dilute area with a lower solute concentration, and it's going to move to the hypertonic area, or the saltier, quote unquote saltier area. It's going to move to the more concentrated area. I'm going to try to see if I can find a good YouTube video. Usually at this point I would kind of go over to the board and kind of walk through, what a solute is versus a solvent, and kind of rehash the concept of tonicity, hypertonic, isotonic, hypertonic, because you really kind of have to understand that principle to understand which way the water is going to move. And I think a lot of times people get hung up in the terminology in terms of high solute versus what the solvent is, and hyper and hypo. So I'll see if I can find a good YouTube video that really covers that. If you're already comfortable with it, great. Just keep on rolling with it. But if you do think you need a refresher on that, then I'll try to come up with something to post so that you can really get that concept. So this is going to kind of demonstrate the movement of the water. Okay, so looking at the top part, when you drink a glass of water, the water is going to enter the blood from your digestive system. It hits your GI tract first, it gets absorbed, and now it becomes part of your blood plasma. So consequently, the plasma osmolarity is going to decrease. Basically, we have sort of diluted the blood plasma. So it becomes hypotonic to the intracellular fluid. So she's drinking the water. Everything was normal. We had like a normal kind of isotonic situation where this was the same as this. But when she drinks the water, it goes into the GI tract and it comes into the blood. So now you can think of this as being sort of dilute. It's hypotonic. So now you have a higher water concentration in the plasma than you do in the cell, or otherwise stated, the cell is hypertonic to the plasma. So the water is going to diffuse out of the plasma, across the interstitial fluid, and then into the cell. So you get this net movement of water going from the blood to the cell. And so even though we didn't have kind of a starting size for the cell, you can imagine that anytime the cell takes on additional water, it's going to increase in size. Not anything major in this case. I mean, we're not going to like rupture the cell or anything just from drinking a glass of water, but just, to state the fact that as you increase the fluid volume of the cell, it's going to enlarge somewhat. Okay, look at the bottom half, and this is going to be the opposite. So she's losing a lot of fluid this way. It looks like she's sweating. So if water is lost from the body and it's not replaced in a timely manner, then dehydration occurs. So we've lost water from the plasma. So now we originally had isotonicity. We had the same amount, or kind of same concentration between the two compartments, but as we lose water from the blood, it becomes hypertonic. So now we have more fluid in the cell than we do in the blood. So that water is going to be free to leave the cell, pass through the interstitial space, and then make its way into the plasma, again, to try to balance the two out, because it always wants to be as equal as possible. So here we've got a net movement of the water moving from the cells to the plasma, and then of course it's got to pass through that interstitial fluid on its way there. So fluid balance exists whenever intake is equal to fluid output. And you've got a normal distribution of water and solutes present in the two major fluid compartments, being intracellular versus extracellular. So all the way we can get fluid intake is through preformed water. So that's basically just drinking water and water that's taken in from our food. That goes into the GI tract. It's about 2,300 milliliters per day. You've got metabolic water, which includes the water that's produced from your daily cellular reactions, most of which are aerobic cellular respiration, and then dehydration reactions. So our cells are doing all their metabolic activities, and they can create water that way as well. So taking it in, and then our cells kind of doing what they do, forming water, that's how we're going to get our intake. Output happens through breathing. We lose some, not a ton, but we lose some when we breathe. Sweat, cutaneous transpiration, which is evaporation of water directly through the skin, and then defecation, urination. So you can see that on the chart. Here's all the ways we're taking it in, and here are the various ways that we can put it out. These are just some definitions associated with the description of water loss. So we can describe water loss in a couple of different ways. You can describe it in terms of being sensible and insensible, which boils down to the ability to measure what's the fluid that's being lost. Or you could do obligatory versus facultative, which is more associated with your state of hydration and whether it's kind of mandatory or more of a regulated water loss. So sensible water loss is anything that's measurable. So any fluid that's lost through the urine and feces is going to be sensible water loss. Insensible water loss is that which you cannot measure. So it's going to be the fluid that gets exhaled in the air that you're breathing, and the fluid that's lost through that cutaneous transpiration or that evaporation through the skin. So there's not really a good way to measure those. So those are considered insensible water loss. Obligatory water loss is the loss that's going to always happen regardless of your state of hydration. So this is still going to happen even if you're in a dehydrated state. So it's going to be the water loss that happens through breathing and through cutaneous transpiration, as well as the fluid that's going to be lost in the feces and that minimal amount of urine that you have to produce to get rid of waste products. So remember I mentioned that even if you're not well hydrated, you have to at least produce half a liter of urine a day to be able to get rid of waste products. So that half a liter is going to be considered obligatory water loss. If you are well hydrated and you're going to produce more urine than half a liter per day, then anything above that would be considered facultative. So facultative water loss is controlled water loss. That's going to be, again, dependent on the state of hydration, and we regulate that by hormones in the distal convoluted tubules and the collecting tubules in the kidney. Fluid imbalances happen whenever you have a difference in the fluid input and the fluid output. We can kind of categorize these in a few different ways. So you've got volume depletion. Volume depletion is when isotonic fluid loss is greater than isotonic fluid gain. So you're losing more than you're putting in. The fact that it says isotonic means we're not changing sodium or electrolyte concentrations here. It's just you're talking about the same osmolarity of the fluids, but you got more going out than you have coming in. So you end up with a depletion. So examples of condition that would cause volume depletion would be hemorrhaging, severe burns, chronic vomiting, diarrhea, or hyposecretion of aldosterone, if you're not making enough aldosterone. So remember, aldosterone moves sodium and water back into the blood. So if you don't have enough of that, it's going to stay in the tubular fluid, so you're going to excrete excess urine. The other one's going to be volume excess, when the isotonic fluid gain is greater than the isotonic fluid loss. So this typically results whenever the fluid intake is normal, but you've got a decrease in fluid loss through the kidneys. So basically your kidneys aren't functioning that well, or you may have a hypersecretion of aldosterone. So you don't really need that much sodium and water being reabsorbed, but for whatever reason aldosterone is being hypersecreted, so you're getting more reabsorption, so you're not getting enough excretion of the fluid with the sodium. So again, note that in both volume depletion and volume excess, there's no change in osmolarity. So when you're talking about the fluid that's coming in and the fluid that's going out, it's the same osmolarity. You just have an unequal distribution of what's coming in versus going out. So the consequence there is there's going to be no net movement of water between the different compartments. We're not creating a hyper and hypotonic solution between, say, the urine and the blood. It's all staying the same. It's just a volume issue there. Dehydration can result from profuse sweating, diabetes mellitus, or hyposecretion, so low levels of antidiuretic hormone. Remember, antidiuretic hormone stimulated water reabsorption in the kidney by adding those aquaporins in. Additionally, it could just be from insufficient water intake. So in any of those cases, you've got water loss is greater than the loss of solutes. So the blood plasma becomes hypertonic. So it's almost like we're pulling just the water and the water alone from the blood, and we're excreting that. And what that's doing is it's sort of concentrating what's going on in the plasma, so it becomes hypertonic. So consequently, the water then shifts between the fluid compartment. You have a net movement of water from the cells into the interstitial fluid and then into the blood plasma. And so the body cells can become dehydrated as a result. So we were losing water, just water alone, that sort of concentrated the blood. And remember back when we looked at the diagrams of kind of where you had the blood and then the interstitial space and then the cells, we kind of want that to stay as even as possible between the osmolarities. So if the blood becomes hypertonic to the cells are going to respond by releasing their water through the interstitial space back to the blood to get it balanced back out. So consequently, then the cells have now become dehydrated. Fluid sequestration is different from the other ones because you don't have a difference in the amount of fluid that's either coming into the body or leaving the body, but rather you just have a distribution issue where the fluid is now distributed abnormally, as you can see in this picture. So fluid will accumulate in a particular location, and so you have kind of excess fluid in one area, which means then you don't have it available for use in a separate area. So edema is an example of fluid sequestration where you get fluid accumulation in the interstitial space surrounding cells in a certain area, and that's going to cause this puffiness or this swelling. Edema is normally a result of abnormal changes in the cardiovascular system, so your heart or blood vessels could also be due to changes in the lymphatic vessels. So the changes alter that net filtration pressure at the systemic capillary, so you have maybe like an increased net filtration pressure, and that's going to cause additional fluid to leave and go into the interstitial space. So you're going to get an accumulation. The alternative is if you have normal filtration, but for whatever reason your lymphatic vessels aren't able to collect that fluid, then it doesn't quite leave the area, because normally you would filter fluid out of the blood vessels, it'll stay in the interstitial space, and it'll either get back into the blood vessels or it will go into the lymphatic system. So if it's getting in that interstitial space and then not going back into the blood vessels or into the lymphatic system, then it's just staying there, and that's going to cause that swelling or that edema. So there's another clinical view. I'm again going to just kind of let you read through this one on your own. Basically, the two populations, or two of the populations that are more highly at risk for dehydration, are going to be infants and elderly. So either really young or really old. Kind of due to some different reasonings. They've got it listed out here. Again, I'm not going to read through those, but I do want you to go through that on your own. And just kind of be familiar with the different reasons why these two groups are so susceptible to dehydration. So in order to maintain fluid balance, we have to kind of monitor both the amount of fluid that comes in and the amount of fluid that leaves. But the thing is we don't have any receptors that directly monitor the actual amount of fluid in the blood or even just the grams of dissolved solute. So we don't have anything to say, okay, we have X amount of sodium directly in our blood. So we kind of have to monitor it through sort of indirect mechanisms. So we can do that by monitoring the blood volume, the blood pressure, and the plasma osmolarity. Okay, so we know that fluid intake increases blood volume. So the additional volume that gets put into our blood whenever we take in increased fluid has two different consequences. One, our blood pressure increases. So as volume increases, pressure increases. And two, if the water gain exceeds the solute gain, then the blood osmolarity is going to decrease. So in case you don't remember what the solute is, just imagine I have a glass of water. The water is going to be what's called the solvent. And then if I start pouring salt into it, the salt is called the solute. And when you mix a solute into a solvent, it creates a solution. So what this one is saying in here is if you're taking in kind of just pure water, so we're getting in more fluid but not any of the solute, we're basically diluting the blood, and so the blood osmolarity will decrease. So on the other side of that, when we have increased fluid output, that's gonna decrease blood volume. So more fluid output results in lower blood volume, and so therefore lower blood pressure. And if we're losing more water than solute, then we're basically concentrating the blood. So that means our blood osmolarity is going to go up, or it's going to increase. So to prevent these imbalances from taking place, our body has to have a way of regulating the amount of fluid intake and fluid output. So this one talks about regulating the fluid intake through activation of the thirst center, which is an area in the brain. The thirst center, when it becomes activated, it causes you to feel thirsty, so then you want to drink and take in more fluids. So this is something that becomes activated whenever you don't have sufficient fluid in the blood. So decreased salivary secretion, so saliva production is going to go down, and the mucous membranes become less moist anytime you don't have as much fluid available. So you got sensory receptors in there that will detect the reduced salivary production in the drier mucous membranes, and that sends signals up to the thirst center, and that makes you feel thirsty. You take in more fluid and try to correct the problem. Increased blood osmolarity, so this happens mostly from either insufficient water intake or dehydration. So if you're not taking enough water in, or if you're getting rid of just water, but all those solutes are staying in the blood, it basically concentrates the blood. And that increase in blood osmolarity stimulates the sensory receptors in the thirst center directly, and it also stimulates those some cells in the hypothalamus to initiate nerve signals that go to the posterior pituitary, and that's, if you remember, where we had antidiuretic hormone, or ADH. So it stimulates the thirst center so that you'll start to take in more fluid, but it also stimulates the hypothalamus to tell the posterior pituitary to release ADH. And ADH increases water reabsorption in the kidney tubules. So if we're in a situation where we have high blood osmolarity, we don't have sufficient water in the plasma. So we're taking it in through this because we've activated the thirst center, but that ADH is going to also move more water from the tubular fluid into the blood. So we're not going to just excrete everything that we take in, but rather save it in the blood. And then also decrease blood pressure. So when fluid intake is less than fluid output, the blood volume is going to decrease, and then that leads to a decrease in blood pressure. Renin gets released from the kidney in response to that lower blood pressure. Renin causes higher levels of angiotensin II, and angiotensin II within the blood can also act to stimulate the thirst center so that, again, you drink and you take in more fluids and bring that blood pressure back up. Stimuli for inhibiting the thirst center occurs whenever fluid intake is greater than fluid output. So now we have sufficient, if not excess, fluid in the blood, so we don't need to be thirsty and continue to take in water. So increased salivary secretions. So when body fluid level is high, salivary secretions increase and the mucous membranes of the mouth and the throat become very moist. So sensory input to the thirst center kind of goes down or becomes halted. Distension of the stomach. Fluid entering the stomach causes it to stretch, and that provides inhibitory sensory impulses that go back to the thirst center. Important to know that an empty stomach does not stimulate the thirst center. So this is kind of a one direction only thing. When the stomach stretches, as it will, as fluid enters the stomach, it basically inhibits those impulses that would otherwise go to the thirst center, so that we kind of we're not activating, or otherwise we're inhibiting the thirst center. But again, it doesn't work in reverse. An empty stomach is not one that's constantly stimulating the thirst center. Decreased blood osmolarity. This is basically like having dilute blood, so you don't need to add additional fluid. So the blood osmolality will decrease as you continue to add additional fluids into the blood. So in response, the thirst center stops becoming stimulated, and the hypothalamus will also reduce its communication with the posterior pituitary so that it will stop releasing antidiuretic hormone. Because if we have sufficient amount of fluids or excess amount of fluids, we don't need the antidiuretic hormone around. We're perfectly fine to excrete all that extra fluid with the urine. And then increased blood pressure. So blood volume and blood pressure, we know, increase as you continue to add fluid into the body. So that increase in blood pressure would inhibit the kidneys from releasing renin, and the subsequent production of angiotensin II is then going to go down. So that angiotensin II stimulated the thirst center, so as it goes down, we'll get a reduction of that stimulation, so that we're not stimulating the thirst center because we don't need to continue to take in additional fluids. Fluid output is regulated through the kidneys by controlling our urine output, so we've kind of seen that. We've got 4 major hormones that are involved in regulating urine output. We have seen all 4 of these before, so nothing really new here. Angiotensin II, antidiuretic hormone, aldosterone, and atrial natriuretic peptide. So I want you to become familiar. You might even make yourself a chart where you write down the 4 hormones, where they come from, or at least in the case of angiotensin II, basically kind of what creates it, because that's the one where you have, it starts with renin and angiotensinogen, and then it leads to angiotensin II. So generally, their name, kind of either where they come from or what, causes their formation, like an angiotensin II, and basically what they do, so that when you see one of these hormones quickly, you can remember, okay, aldosterone came from the adrenal cortex, and that one was an increased sodium channels in the renal tubules. Okay, so angiotensin II, antidiuretic hormone, and aldosterone, all 3 help to decrease urine output. All 3 of them function to maintain both blood volume and blood pressure. ANP is the only one that kind of does the opposite. ANP increases urine output, and it decreases both blood volume and blood pressure. So whenever your blood pressure or your blood volume is high, ANP is going to be the hormone that goes and it increases the amount of fluid that gets excreted with the urine, so it pulls fluid out of the body so you can get your blood volume and blood pressure back down. Okay, the specific mechanisms employed by each of these hormones in regulating fluid output in the kidneys also function to regulate some of the electrolytes. So, classic example of that is aldosterone. We have seen that aldosterone ultimately causes an increase in water reabsorption, but it does it through an indirect mechanism. The direct mechanism is that it increases sodium channels in the kidney tubules. And so basically, even though it's regulating water, it's also regulating sodium. So let's look at the difference between electrolytes and non-electrolytes. Non-electrolytes are molecules that do not dissociate or come apart when they get into solution. Most of these are going to be covalently bonded organic molecules, such as glucose. So if I have a glass of water and I have some molecules of glucose, when I put the glucose into the water, it's going to make a glucose solution, but the actual chemical structure of glucose stays the same. It's going to be the same inside the water and outside the water. It's not going to break apart into its different atoms. It stays a cohesive glucose molecule. An electrolyte is a substance that dissociates or comes apart when it gets into water. And when it comes apart, it's going to form cations and anions. So, refresher, positively charged ions and negatively charged ions. So electrolytes are going to be your salts, your acids, your bases, and sometimes negatively charged proteins. So sodium chloride is a salt. It's one cohesive molecule, but as soon as you put it in water, it breaks apart into a sodium cation and a chloride anion. Calcium chloride, same thing. Put it in water, it's going to break apart into a calcium cation and two chloride anions. Hydrochloric acid, together, when it's by itself, they're attached. When you put it into water, it separates into a hydrogen ion and a chloride ion. This hydrogen ion is what's going to be responsible for causing the pH of a substance to go down, making it acidic. A base, sodium hydroxide, you put it into water and it's going to break apart into sodium and a hydroxide ion. So electrolytes, they're atoms that are combined together, but as soon as you put them in water, they break apart. Non-electrolytes are atoms that are combined together. You put them into water and they stay together. Some of the common electrolytes that we have in the human body, we've got sodium and potassium. These guys are cations because they're positively charged. Anions, you've got chloride and bicarbonate. They have a variety of functions. A lot of these function in ways that we've already seen in 2401 and 2402. We're going to continue to talk about additional functions. But to carry out the functions effectively, we have to maintain each electrolyte concentration within this kind of normal homeostatic range. So each one has its own range, and we have to make sure that, their levels stay in that range so everything can kind of do its job the way it's supposed to. So we'll start by just looking at sodium ion. 99% of sodium is in the extracellular fluid, and only 1% of it's going to be in the intracellular fluid. And again, that comes back to the sodium-potassium pump. So all of the cells in our body have the sodium-potassium pump, and its only job is to continually pump sodium out of the cell and potassium into the cell. So sodium is going to be the principal cation, positively charged ion, in the extracellular fluid. Normally it's present in one of two different forms, either a sodium bicarbonate, which is NaHCO3. So remember when we saw that HCO3 minus, I said that's bicarbonate. You just attach the sodium, and now you have sodium bicarbonate or sodium bicarb. Sodium chloride, NaCl. So sodium exerts the greatest osmotic pressure in the extracellular fluid, meaning it's the main factor that draws fluid to that area. I've said it before, wherever sodium goes, water likes to go. So if you have an area that's very highly concentrated in sodium, it's got a great osmotic pressure that pulls fluid into it. Sodium has a lot of physiological functions. We've talked about some of them. Neuronal excitation. I think that was at the end of 2401 when we talked about how neurons fire in the nervous system. Glucose co-transport in the kidneys. We saw that in the urinary system when glucose got filtered, and then we had to reabsorb it, and we had to move that glucose against its concentration gradient from the tubular fluid into the cell. And it did that by traveling alongside sodium through that transporter, where sodium was moving with its concentration gradient. We get sodium from our diet. It comes primarily from table salt and processed foods. We lose it in the urine, the feces, and the sweat. And then the amount of sodium that's lost in the urine is typically regulated by 3 different hormones: aldosterone, antidiuretic hormone, and atrial natriuretic peptide. So you should be familiar with by now with kind of how this works. how these work. If you're not, you may want to go back and rehash that. So this is basically just kind of a summary of what that last slide said. Here's your normal sodium balance. Your normal sodium levels. I would know these numbers. I didn't type them in on my slide. I used to have them and I deleted them. Since we're going off of Connect exams now, instead of, because that is not a question that I would have written into one of my exams, but it's very possible for a Connect exam. So I would make sure that you probably commit to memory the normal range for all of these electrolytes. Okay, so sodium is going to be between 135 and 145 milliequivalents per liter. That's the unit for these, milliequivalents per liter. This tells you how we take it in. We take it in through our diet. We put it out with our urine, our feces, and our sweat. And then these are the hormones that regulate sodium concentration, either by altering the loss of sodium or water or both. So aldosterone retains sodium and therefore water. When I say retains, we're moving it from the tubular fluid back into the blood. ADH retains water. So ADH is not going to directly affect the movement of sodium. It's not going to directly affect sodium reabsorption or anything like that. But if you have a certain amount of sodium in the blood and then ADH comes out and it causes more water to come into the blood, what we've done is sort of diluted our blood, or we've decreased the sodium blood plasma concentration. And then A and P is the one that increased glomerular filtration rate, which means everything speeds through the tubular system and we end up excreting more sodium and more water in the urine, which is going to reduce the amount then that's in the blood. Sodium is going to be the most important electrolyte in determining blood plasma osmolarity, which means that it's going to be the most important electrolyte in regulating fluid balance. So the extracellular fluid, and here I'm speaking primarily about the plasma, is going to become temporarily hypertonic to the intracellular fluid if sodium concentration increases from either an elevated sodium content, if you ate like a really sodium-rich meal, or decreased water content if you're not taking in a lot of water. So basically, if you're taking in too much sodium and not enough water, you're going to elevate the sodium concentration in the plasma, which is part of the extracellular fluid. So if you do that, it becomes hypertonic to the cells. So what's going to happen then is water is going to move from the intracellular fluid, or it could also move through the interstitial fluid, into the blood plasma in an attempt to kind of normalize or equalize the concentrations. So the reverse can be true. Again, the extracellular fluid, mostly speaking of the plasma, can become temporarily hypotonic to the intracellular fluid if sodium concentration decreases, either as a consequence of taking in less sodium or taking in a lot of water. So let's just say you start to drink a ton of water, pure water, then that's going to go through the GI tract into the plasma, and it's basically going to dilute the blood plasma, which means that component of the extracellular fluid has become now hypotonic to your cells. So in that case, the water is going to move from the plasma into the cells to reestablish that normal sodium concentration. Changes in plasma volume directly influence both blood volume and blood pressure. We've seen that. As volume increases, the pressure is going to increase. Sodium retention leads to water retention in the plasma, which leads to high blood pressure. So the more sodium that we take in through our diet, that means the more water. So water follows sodium. So the more sodium in the plasma, the more water in the plasma. That's going to lead to high blood pressure. Okay? If we have a loss of sodium, water's going to follow. So we're going to get a loss of water from the plasma. That's going to translate to low blood pressure. So this is why clinicians tell patients with high blood pressure to limit their sodium intake. If they're already battling a high blood pressure and they continue to take in high levels of sodium, that means they're going to continue to retain large amounts of water. So now they have a higher blood volume, and it's going to lead to even a higher blood pressure. So by limiting that sodium intake, we limit that fluid retention, and we can start to kind of at least maintain the blood pressure, if not get it lower and lower. So this is a diagram of what I was just talking about with the movement of sodium. I'm sorry, with the movement of water as a result of sodium. Okay, so this was the first case scenario. This top box is what would happen if you had an increase of sodium consumption or a decrease in water consumption. So basically you've got a lot of sodium in the plasma and less water. So what that does is it creates a difference in between the concentration of the blood and the cell. So in this case, the blood is hypertonic because there's more sodium concentrated in the blood. So it's hypertonic to the cell. So the water is going to flow out of the cell into the blood to kind of try to dilute that, to try to get back to a balance of where it needs to be. The bottom panel describes the second scenario that we discussed, where if you have a reduced sodium concentration, so if you just start drinking a whole bunch of plain water, it's going to kind of dilute the sodium in the plasma, so now you have a reduced sodium concentration. So you're going to leave, the water is going to leave the plasma and go into the cells to try to reconcentrate the sodium back to the concentration that it needs to be. And you can see that this is directly going to affect the size. It's not really demonstrated in the picture, but as fluid leaves the cell to go into the plasma, the cell is going to shrink in size. As fluid leaves the blood to go into the cell, it's going to cause the cell to grow larger because it's filling with fluid. Sodium imbalance is one of the most common types of electrolyte imbalances that you find. Hypernatremia is where it gets too concentrated in the blood. Hyponatremia is where it gets too dilute in the blood. So hypernatremia, your sodium levels are above normal. It can be caused by dehydration or diabetes insipidus. So we'll talk about diabetes insipidus later, but basically that's what happens when you get an inadequate secretion of antidiuretic hormone. So you don't have that hormone there to promote water reabsorption, and so that leads to an increase in water excretion in the urine. So if you're losing that much fluid in the urine, it basically concentrates the levels of sodium that are in the blood. So you end up with elevated sodium plasma levels, and then the consequences is that the cells in the body are going to try to compensate for that by donating their water, their fluid, so you get water leaving the cells and going into the plasma to help reestablish the balance, and so as a consequence of that, the cells will shrink. Hyponatremia is when you get sodium levels that are below normal. It can be caused by excessive water intake. So if you're just drinking large amounts of pure water, it's basically going to kind of dilute the blood, so it dilutes out that sodium concentration. Hypersecretion of antidiuretic hormone. So if you have a lot of ADH floating around, you're basically going to reabsorb all of the possible water in the tubular fluid and move it back into the blood. So again, you get a very dilute blood plasma, and that's going to lower your sodium concentrations. Diarrhea, burns, and excess sweating can all also lead to lower than normal sodium concentrations in the blood. So the consequence here is that we're going to move some of that excess fluid that's in the blood into our cells. So you get swelling of the cells, and then it results in a decreased blood volume and blood pressure as we move that fluid out of the plasma. So in contrast to sodium, the majority of potassium is going to be found inside the cell. So about 98% of potassium is found in the intracellular fluid, with only 2% of it found in the extracellular fluid. And again, that's because of the sodium-potassium pump. So again, all of our cells have the pump. They're constantly pumping sodium out and potassium into the cells, so potassium is accumulating in the cells. So potassium is going to be the principal cation to exert intracellular osmotic pressure because it's going to be the most abundant cation inside the cells. Potassium has a very important role in controlling the heart rhythm. So although the vast majority of the potassium is located inside the cell, it's actually only that minimal amount, that 2% of the potassium that you find in the extracellular fluid that's actually continually regulated. So very small changes in blood potassium levels can very quickly lead to a potassium imbalance, and potassium imbalance is going to be the most lethal of the electrolyte imbalances. And that is because potassium has such a significant role in regulating the rhythm of the heart. If it gets out of balance, then it can mess with the heart very quickly and lead to either cardiac or respiratory arrest. Potassium is generally obtained from fruits and vegetables. Only a small amount of potassium gets lost through the sweat and the feces. The majority of potassium gets lost in the urine. So here's that chart again for potassium. So this basically has, for the most part, has almost all of the information that was in the previous slide. Again, here's our potassium levels that we should see. Again, I would make sure to probably commit these to memory for each of the different electrolytes or ions. Potassium intake comes from our diet, from fruits and vegetables. We're going to lose most of it in the urine, but we do lose some in the feces and the sweat as well. Hormones that regulate potassium is going to be aldosterone. So if you remember aldosterone, again, the primary action of aldosterone was to increase sodium reabsorption because it increases sodium channels. But we talked about how in the distal convoluted tubule and the collecting tubule, because of the sodium-potassium pump, if you're moving sodium from the tubular fluid back into the blood, because of that pump, it moves sodium one way and potassium the other. So that means as sodium's going into the blood, potassium's going from the blood into the tubular fluid, which is going to become the urine. So you're actually getting potassium secretion, and you're finding potassium getting excreted into the urine, or getting excreted with the urine. So that's going to cause a reduction of potassium in the blood plasma concentration. So these are just some of the things that can kind of affect the movement of potassium. So this top one is just kind of a common sense kind of thing, maintaining normal blood potassium levels. So if the blood potassium level increases, so if we have too much potassium in the blood, it's obviously going to move into the cells, because we got to keep our blood potassium in a very specific range that I showed you on the other slide. And then if it starts to go down, we'll just move some of the potassium out of the cell and into the blood to kind of reestablish those normal levels. It can also move in between the blood and the cells based on pH. If the blood hydrogen ion increase, this causes the blood to become acidic. So the hydrogen ions they need to get out of the blood, so they'll start to enter the cells. And there's kind of a similar anti-Porter situation that you have with potassium and hydrogen, like you had with potassium and sodium, where as hydrogen moves into the cell, potassium moves out of the cell. So if the blood pH starts to go down because we're getting acidic, we're going to basically move some of those hydrogen ions out of the blood and into the cells. And as the hydrogen moves in, the potassium moves out. So you'll see an increase in potassium in the plasma. The opposite occurs if the blood starts to become alkaline. So the pH is going up in this case. We're right here. If the blood concentration of hydrogen ions goes down, we need to get more hydrogen ions in the blood. So hydrogen is going to come out of the cell and go into the blood. But because of that anti-Porter that's there, as hydrogen comes this way, potassium is going to go the other way. So you're going to get a potassium movement from the plasma to the cells. So you're going to end up with a decrease in plasma concentration during alkaline conditions. And then the last one talks about the movement of potassium that can happen as a result of glucose movement. So after you eat a meal, all the glucose goes in from the GI tract into the blood. So now we've got a lot of glucose in the blood. Glucose is going to move out of the blood and into the cells. We're familiar with that because of insulin. And potassium is going to basically go with it. It's going to follow. So insulin increases not only the movement of glucose from the plasma into the cells, but also the movement of potassium. So you see them both going out of the plasma into the cells. So just a couple of additional conditions that can influence the movement of potassium between the cells and the plasma. Chloride is a common anion, so it's got the negative charge, and it's normally associated with sodium. So sodium chloride, you always see those two together because they've got a really good electrostatic interaction. So chloride's going to be the most abundant anion in the extracellular fluid. Just like sodium was the most abundant cation in the extracellular fluid, chloride's going to be the most abundant anion there as well. You also find it in the lumen of the stomach because we had a bunch of that hydrochloric acid. You remember that was getting released by the parietal cells in the gastric pits, if any of that rings a bell. So you got a lot of HCl going on in the stomach, so you find a lot of chloride there. We get chloride mostly from the diet. It's going to come mostly from table salt and then processed foods. And then we can lose it through the sweat, those stomach secretions, and also the urine. The amount of chloride that gets lost in the urine is going to be primarily dependent upon your blood plasma sodium levels. So because chloride likes to kind of hang along with sodium, all of the mechanisms that we use to regulate sodium reabsorption, chloride kind of follows the same pattern. We also use those to regulate chloride movement because it likes to move along with sodium. Calcium is the most abundant electrolyte in bone and teeth. So calcium likes to kind of crystallize with phosphate salts to kind of create this mineralization effect that we can use to deposit that into the more solid structures such as bone and teeth. So about 99% of all of the body's calcium is stored in the extracellular matrix of either bone or teeth, and that again causes them to harden. To prevent hardening or calcification of other tissues, calcium is usually moved by calcium pumps either out of the cells or into specialized calcium storage organelles such as sarcoplasmic reticulum. So because calcium does have such a high tendency to harden, we don't want it just creating these mineralizations inside of our cells. That would be very bad. So we either constantly pump it out of the cells before it can harden, or we keep them tucked away in these specialized structures. I always refer to them as calcium closets, where we just kind of put them until we need them. So the sarcoplasmic reticulum, that, if you remember from muscle tissue, was basically where we stored the calcium in it until it was time for muscle contraction. Okay, so in addition to hardening the bones and the teeth, calcium is needed to initiate muscle contraction. Everybody remembers that from 2401, hopefully. And it was also used to release neurotransmitter. That was also from the very end of 2401. Calcium came into those axon terminals and allowed those vesicles to fuse with the plasma membrane, which released the neurotransmitters. It also serves as a good second messenger. So we saw that this semester at the very beginning in the endocrine system with the phospholipase C pathway. If anybody still has nightmares from that, but one of the things it did was cause calcium release, and then calcium went off and served as a second messenger. And then it also participates in blood clotting, which we kind of skipped over a little bit this semester. So it's a very important ion. It has a lot of different functions. We get it from our diet, primarily in yogurt, milk, soy products, cheese, sardines, and those leafy green vegetables such as broccoli and collard greens. And then we lose it in the urine, the feces, and the sweat. So now we're going to look at the hormonal regulation of some of these fluid and electrolytes with our favorite 4 hormones: angiotensin II, antidiuretic hormone, aldosterone, and atrial natrodipeptide. Okay, so let's start with angiotensin II. You guys know I love these diagrams because I think that they're very thorough, and I think it does a good job allowing you to move from the stimulus, kind of in a stepwise matter all the way to the effects, which are in the blue boxes. Okay, so if we have a low blood pressure, or if we get activation of the sympathetic nervous system, that's going to be the stimulus. You've got juxtaglomerular cells and the juxtaglomerular apparatus. I'm not going to do that. The next hormone to look at is antidiuretic hormone, or ADH. So here's some of the things that can cause its release. Angiotensin II can cause a release of ADH, low blood volume, or increased blood osmolarity, meaning increased blood osmolarity means we have sort of concentrated the blood. We've lost water from the blood. So we have low water concentration, but maybe a high solute concentration. So the hypothalamus kind of responds to that. You can see here the hypothalamus sends signals down to the posterior pituitary. So if you remember when we talked about the endocrine system, ADH was synthesized in the hypothalamus, but then it moves down and is stored in the posterior pituitary. So when the hypothalamus receives those stimulus signals, it sends messages to the cells of the posterior pituitary, and they release antidiuretic hormone. So antidiuretic hormone acts on the hypothalamus to activate the thirst center. So it makes you feel thirsty. You increase your fluid intake, that increases your blood volume and increases your blood pressure. It also acts in the kidneys. We talked about this in the last lecture. It increases water reabsorption. So it puts more of those aquaporins on the cells of the distal convoluted tubule, collecting tubules, collecting ducts, kind of that terminal portion of the tubule system. And then more water can go from the tubular fluid back into the blood. So it increases the reabsorption of water. And then at really high levels of ADH, it can cause blood vessel vasoconstriction. So that's not its primary job, but if ADH is around for long enough and high enough concentrations, it can lead to vasoconstriction, which would increase blood pressure. So here's a clinical view for diabetes insipidus. So this is the one we talked about earlier, and so we were going to talk about it in detail later. So despite the fact that diabetes insipidus and diabetes mellitus both start with the same word, completely different diseases, completely different issues. We know diabetes mellitus is an issue with glucose uptake, usually a result of insufficient insulin production. Diabetes insipidus has to do with antidiuretic hormone. So it can happen for one of two different reasons, but ultimately it kind of translates into the same thing. One is either hyposecretion of ADH from the posterior pituitary. So the posterior pituitary is just simply not releasing enough ADH. Alternatively, the kidneys are just not responding to the ADH that's there. So even if the posterior pituitary is releasing the proper amount, if the cells in the kidneys don't listen to it and don't respond to it, then it doesn't do any good. So in either case, you're just not getting a signal for ADH in the kidney tubules. And remember what ADH does is it causes the reabsorption of water from the tubular fluid into the blood so that we don't excrete so much water in our urine. So if you have diabetes insipidus, it leads to a decrease in fluid retention, so we're not pulling that fluid from the tubular fluid into the blood, and an increase in urine production. So these people can lose up to 20 liters of fluid per day because whatever fluid gets filtered is essentially the same amount of fluid that's going to end up getting urinated out. So they lose vast amounts of urine or fluid in their urine daily. The next one is aldosterone. So here are some of the stimuli for aldosterone. Angiotensin II can cause aldosterone release, decreased sodium blood plasma levels, or increased potassium blood plasma levels. So aldosterone, we also remember from the endocrine section, came from the adrenal cortex. So the adrenal cells in the adrenal cortex respond to any one of these stimuli and they release aldosterone. Aldosterone is going to travel down to the kidneys, and this is the one that put more sodium channels on the tubule cells. So it increases the reabsorption of sodium, so you have sodium moving from the tubular fluid back into the blood. Look at water, it's doing the same thing, but it's doing it just because it likes to hang out with sodium. So because sodium moved across, Water's going to do the same thing. So water's moving from the tubular fluid into the blood. Now, because of the sodium-potassium pump, if sodium goes into the blood, that means potassium is going to go from the blood into the tubular fluid. So it's increasing potassium secretion. So aldosterone, like technically what it does is increases sodium channels in the tubule system. The effects that it has are an increase in sodium reabsorption, which increases the sodium amount in your blood, an increase in water reabsorption, which increases the water in the blood or otherwise can reduce the osmolarity, and it increases potassium secretion, which means potassium is moving out of the blood and into the tubular fluid, and there it's going to get excreted. And then atrial natriuretic peptide, or ANP, this is the one that's different from all of the others because this is the only one that we really talk about that doesn't have to do with increasing blood volume, increasing blood pressure, and decreasing urine production. All the other three, angiotensin II, aldosterone, and antidiuretic hormone, they do just that. Those increase blood volume, they increase blood pressure, and they decrease urine production. ANP does the exact opposite. So this is the guy that acts differently than everybody else. So atrial natriuretic peptide, as the name suggests, comes from the atria and the heart. And it gets stimulated to get released whenever you get increased stretch in the baroreceptors in the atria. So basically, if you have too much, I shouldn't say too much, if you have an increase in volume of blood that's coming back to the atria, the right atria, it starts to stretch that chamber. And baroreceptors respond to stretch. So when those baroreceptors become activated as it stretches, it releases atrial natriuretic peptide, or ANP. So some of the things that ANP does, it causes vasodilation. So those blood vessels will vasodilate, they'll open up. So that provides kind of a quick fix to kind of combat what was happening here. Basically, what was going on in the atria is you were getting this really fast return of a large volume of blood, and the atria can only accommodate so much blood. So if you vasodilate your systemic blood vessels, it allows more room for them, and then they can stay there longer. They don't have to immediately rush back to the atria. So it kind of releases some of the demand that's happening in that collection chamber. Okay, so vasodilation of systemic blood vessels, that's going to decrease peripheral resistance, and it's also going to decrease blood pressure. It's going to go down to, it's going to act at the kidneys where it increases glomerular filtration rate, or GFR. So remember, I said anytime we increase glomerular filtration rate, that's going to increase the amount of urine that gets produced. And so we're losing more fluid in the urine, so it reduces blood volume and it reduces blood pressure. It increases the loss of sodium and water in the urine, again decreasing blood volume and decreasing blood pressure. Decreased release of renin, and that leads to decreased release of aldosterone and antidiuretic hormone. Okay, so changing topics a little bit. Everything from the rest of this lecture is going to have to do with acid-base balance, or pH balance. So just kind of some reminders here in this slide. Acid-base balance requires the regulation of hydrogen ion concentration in our body fluids in order to maintain a slightly alkaline arterial blood pH. Slightly alkaline because 7 is neutral, everything below 7 is acidic, everything above 7 is alkaline, and our blood pH has to be between 7.35 and 7.45, so it's just a little bit alkaline. We're regulating hydrogen ion concentration because those are the ions that are going to affect pH the most. The more hydrogen ion concentration you have, the lower the pH is going to be, the more acidic it's going to be. Okay, so just like other electrolytes, to maintain the balance, you have to have kind of an equal intake and output. But there's some different circumstances to consider when we're talking specifically about hydrogen ions. So this little reminder here, I just kind of said it, but we'll say it again. As a reminder, pH is inversely related to the hydrogen ion concentrations. So the more hydrogen ions you have, the lower the pH. The lower the pH, the more acidic it is. When you have fewer hydrogen ions present, the pH goes up and it's more alkaline. We have a couple of different categories of acids. We have fixed acids and volatile acids. Fixed acids are acids that are produced from metabolic waste or metabolic reactions. Examples of some fixed acids that we find in the body are lactic acid. Lactic acid comes from anaerobic cellular respiration, or the production of ATP from glucose in the absence of oxygen. That's what anaerobic cellular respiration is. We have phosphoric acid. Phosphoric acid comes from nucleic acid metabolism. So if you remember, our nucleotides have phosphate groups, and so when we digest those, one of the byproducts can be phosphoric acid. And then keto acids come from the metabolism of fat. Okay, so all of our fixed acids get regulated by the kidney, and they do that through the reabsorption and the elimination of bicarbonate, HCO3-, and hydrogen. The other type is the volatile acid. Volatile acid specifically refers to something called carbonic acid. Carbonic acid is produced when carbon dioxide combines with water, as shown. So we saw this before, and I told you you didn't need to know it then, but it would certainly be helpful because I said it was coming back. So it's back. So let me read through this last part, and then I'll kind of walk you through this chemical equation here. Okay, the term volatile here, volatile acid, refers to the fact that the carbonic acid gets produced from a gas that's normally expired, so our CO2. It's regulated by the respiratory system through regulation of the respiratory rate. Okay, so fixed acids come from metabolic reactions or metabolic waste products, and they're regulated by the kidney. Volatile acids come from gases that we exhale, specifically CO2, and they're regulated by the respiratory system by regulating our rate of respiration. So let's see if we can kind of get comfortable with this chemical reaction. And then if you need more time, definitely kind of pause this and, either just sit and think or, look through the textbook or hit up YouTube or something so you kind of understand what's going on. So in a chemical reaction, a lot of times you see an arrow with just one head on it, not a head on the side and a head on the side. When it's just one head, that means whatever on this side of the arrow is going to go to become this thing, and then it's going to stay there and never go back the other direction, which would mean that this could never go back to this. But you'll notice here, all of the arrows in this reaction have two heads. That means that everything can move in both directions. So if you look, there's the same number of atoms. We have what? 1 carbon, 1, 2, 3 oxygens, and 2 hydrogens. Okay? 1 carbon, 3 oxygens, and 2 hydrogens. 1 carbon, 3 oxygens, 2 hydrogens. So we're not losing any atoms. We're rearranging them. We're changing the way that they're interconnected. So here we have carbon dioxide and water. Those can combine into one large molecule, which becomes H2CO3. This is called carbonic acid. If you have carbonic acid, it can do one of two things. It can dissociate just like the way it was and go back to becoming carbon dioxide and water. Alternatively, you can take one of these hydrogen ions and pop it off, and then you end up with this. A hydrogen ion, and this is called bicarbonate, HCO3-. Okay, and all of these can go back and forth as needed. These can go back to this, which can go back to this, or they can just hop back and forth. Okay, so we'll see this kind of come back into play. So I apologize for the amount of text on this slide. I tend not to do that, but sometimes there's kind of no way around it. So this is talking about the regulation of fixed acids by the kidneys. So there's two main sources that we get fixed acids from. One is through our diet, nutrients absorbed in the GI tract, and then the other one is from our body cells, those metabolic waste products. Typically, more acid tends to be absorbed from the GI tract because anybody that eats more of a Western style diet tends to consume a lot of animal protein and wheat, and both of those contribute a lot of hydrogen ions to the blood. And then your cells, of course, are contributing acid waste products as well. So that's that lactic acid, the phosphoric acid, and the keto acids that I talked about. Okay, so acidic conditions can also result from the excessive loss of bicarbonate, which is a weak base, and we'll kind of touch on that here in a little bit as well, as a consequence of diarrhea. So bicarbonate is normally lost in the feces, but if somebody has diarrhea and they're losing it kind of abnormally, then you can lose excessive amounts of bicarbonate. So this top portion is basically telling you different scenarios that can lead to acidic conditions. We can either, get a lot of acid through our diet, a lot of acid through our metabolic waste products, or we had an excessive loss of bicarbonate, potentially through something like diarrhea. Okay, so here's how the kidneys can help reverse excessive plasma hydrogen levels. So for one, they can reabsorb all of the bicarbonate that gets filtered. So we saw that in the urinary system where we freely filtered bicarbonate, but we didn't filter very much hydrogen. So we had to kind of reabsorb, although if you remember, it was very convoluted. It wasn't a straight reabsorption. We had to kind of do a little chemistry there, and I called it reclaim. So we kind of reclaimed all of that bicarbonate. Because basically, when this HCO3 minus comes into contact with a hydrogen ion, the two are going to bind together. And if that hydrogen ion binds to bicarbonate, it's now a neutral molecule. So it stops kind of contributing to the acidic environment. Okay, so the kidneys can reabsorb all of the bicarbonate, and that's going to help to kind of neutralize things. It can also make new bicarbonate while secreting hydrogen into the fluid, tubular fluid by type A intercalated cells. That's you're going to find all of that in the distal convoluted tubule. So think about this second bullet point right here. If you make more bicarbonate, then you can kind of push that off into the plasma, and that can kind of neutralize a lot of the hydrogen there. But if you're also secreting hydrogen ions, that means you're pulling hydrogen out of the blood and putting it into the urine. So that kind of helps you keep the blood pH from getting too acidic. And that's going to be your type A intercalated cells in the distal convoluted tubule. Okay, kidneys can help reverse alkaline conditions. So alkaline is going to be just the opposite of the acidic conditions. So by either not reabsorbing all of the bicarbonate along the tubule, so this means we're going to actually excrete some of this stuff in the urine, or again secreting bicarbonate from the blood into the filtrate. So if it was already filtered and then reabsorbed, but the conditions are alkaline and we need to kind of make them a little more acidic, we can secrete this. We can move it back from the blood into the tubular fluid and excrete it with the urine. While we do that, we can also reabsorb hydrogen ions. So any hydrogen that did get filtered in the tubular fluid, if our plasma is alkaline and we need it more acidic, we have cells that are called type B intercalated cells, and those can reabsorb hydrogen ions into the blood to help get it more acidic where it needs to be. I hesitated to say that because I don't want to say it needs to be acidic, but if we're going alkaline, we need to move it back to neutral. So by reabsorbing hydrogen ions, we're going to get it closer to neutral. So these are some good kind of charts to show you some of the contributing factors and some of what the body would ultimately do to fix it. This one has to do with increased blood hydrogen ion concentration, so a decreased pH, and this is a decreased blood hydrogen ion concentration, so an increased pH. So the top portion shows you all the different things that can contribute to that change in pH. So all of the things that might lead to high hydrogen ion concentration and low pH. Okay, so for one, certain foods. We talked about wheat and animal proteins contribute a lot of hydrogen ions into the blood. So that's what this would be coming from. Cellular waste products, lactic acid from anaerobic cellular respiration, phosphoric acid from nucleic acid digestion, and keto acids from fatty acid metabolism. So metabolic reactions going on here, their waste products can contribute acidic products into the blood. And then we talked about losing bicarbonate and diarrhea. So it's kind of going the reverse direction, but that's because we're losing bicarbonate as opposed to directly adding hydrogen ions. So all of these scenarios contribute to an increase in hydrogen ion concentration in the blood. Everything in the bottom is basically our balancing mechanisms. So if we've already kind of contributed to acidic conditions in the blood, what is our kidney going to do to kind of correct the issue? And it kind of lies it out right here for you. So excess hydrogen ion is going to get excreted in the urine. So we can move some of this hydrogen out of the blood. We're going to put it in the tubular fluid. And if you want to look at it up close, you can see that here. So this is secretion. Hydrogen, because remember, a lot of hydrogen doesn't get filtered. So it'll go through the glomerulus just fine. And here's our, it's going to come out the efferent arteriole and then kind of travel through the peritubular capillaries. So presumably this is a peritubular capillary. It can secrete hydrogen and put it into the tubular fluid. So this is going to get excreted. And then we can reabsorb bicarbonate. So we're putting more bicarbonate in the plasma, and that's going to help kind of absorb some of the hydrogen ions that are already there and help to neutralize it. So this is happening in our type A intercalated cells. Okay, so contributing factors and balancing mechanisms. And this is basically what I said on the slide just before this, but this is what kind of where it's all combined in kind of a picture format. Okay, moving over here. These are contributing factors to a decreased blood hydrogen ion concentration or an increased blood pH. So vegetarian diet or antacid. So if you're not eating a lot of animal protein or you're taking a lot of antacids, you're not getting a lot of hydrogen ion contribution from the diet, into the blood. And the other one is vomiting. So by vomiting, you lose a lot of hydrogen ions. And so any of these cases can make the blood plasma have a higher pH because you're not getting as much acid or you're losing acid. Okay, so the balancing mechanism is basically going to be the reverse of what we just saw. Excess bicarbonate can be excreted in the urine and hydrogen can be added back to the blood, in this case through type B intercalated cells. So if you look really closely at this and you see the direction of the movement, hydrogen's going from the blood to the tubules, to the tubular fluid, bicarbonate's going from the tubular fluid to the blood. This is type A intercalated cell. Type B is doing the reverse. Hydrogen is moving from the tubular fluid to the blood, and bicarbonate is moving from the blood to the tubular fluid for excretion, type B intercalated cells. So this is a really useful clinical view. It talks about how vomiting and how diarrhea affect the hydrogen ion concentration in your blood. Because if you think about it, vomiting and diarrhea, those are associated with your GI tract. So this kind of makes the connection between changes in the GI tract and your plasma concentration. Okay, so looking at vomiting. Vomiting, we know, is the ejection of the contents from the stomach. We also know that in the stomach, there's a lot of hydrogen from that hydrochloric acid. So you end up losing a lot of hydrogen ions when you vomit. So just like we saw in the kidneys, we have cells in the stomach that are kind of exchange. So with those intercalated cells, remember we had hydrogen move one way and bicarbonate move the opposite direction. We have something very similar in the stomach. So if we lose a bunch of hydrogen from vomiting, we need to replace that hydrogen in the stomach. So we start pumping hydrogen in, right? But if hydrogen's going into the stomach, what's coming out? What's got to be going the other way? It's got to be that bicarbonate. So you get a lot of bicarbonate being added to the blood, and the addition of that bicarbonate, as it says here, increases the blood pH. Okay? If it's severe enough, it can result in something called metabolic alkalosis that we'll talk about shortly. Okay, diarrhea is the excessive amount of watery stool. We know that. I already talked about how we can lose bicarbonate. We typically lose bicarbonate in the stool, but if we are suffering from diarrhea, then you're losing it at a much higher rate, and so you're kind of depleting your bicarbonate sources. So again, just like in the stomach, we've got some cells that look very similar to those cells we saw in the kidney. So if we have lost a lot of bicarbonate, we need to replace it. So we start moving bicarbonate back into the intestines. But if bicarbonate is going from the blood into the intestines, what's got to be going the other way? Hydrogen. So we start pumping hydrogen back into the blood. Bicarbonate's going into the GI tract, hydrogen's going into the blood. So we get this increased transport of hydrogen into the blood. That's going to lead the blood to decrease in its pH because you're just adding a lot of hydrogen. Severe cases of that can result in something called metabolic acidosis. And again, we're going to get to that. But it's because of those cells that transport bicarbonate and hydrogen at the same time, but in different directions, that allows changes in the stomach with vomiting and changes in the intestines with diarrhea to lead to pH changes in the blood. Okay, so all of that was fixed acids, mostly those metabolic acids. But I talked about, that we have these volatile acids, and volatile acids have to do with the rate of respiration. So like the kidneys, the respiratory system is going to serve as this physiological kind of buffering system to maintain that acid-base balance. But in this case, it does so specifically by regulating those volatile acids or the carbonic acid. So H2CO3 is the carbonic acid. So blood pH depends on respiratory rate in the following way. Okay, so think about, we know that CO2 is something that gets exhaled. So this is, coming from a respiratory system. Here we see hydrogen and bicarbonate. We know these play a role in pH. So let's read through the following. Increasing the respiratory rate. Okay, so this causes elevated levels of CO2 to be expired. So think about, you breathe in oxygen, you breathe out CO2. So if you are breathing at a rate or a depth that's kind of above what you need, you start to exhale a lot of CO2. Okay, so this results in decreased blood CO2 concentration. So PCO2 in the blood decreases. Let me tell you the actual scientific physiological way that this works, and then I'll tell you the cheat sheet version. So all of these things in this reaction want to stay balanced and equal. If you had a whole bunch of CO2 and very little, bicarbonate, it's not going to stay that way. What would happen is anytime you get a buildup of one of these reactants, it drives the equation this way because they want to keep them even. So then you would get roughly equal amounts of carbonic acid and equal amounts of, these products over here. So if you get a buildup of CO2, it's the way they describe it is it drives the equation to the right, meaning we're going to go this way and we're going to make all these other things. If you get a depletion in CO2, so this goes down, it's going to do the reverse. We're going to drive the equation to the left. We're moving this way because we want to get these levels back up. Okay, so look at that in relationship to what I just said. We're breathing at a really fast rate. We exhale a lot of CO2. So our blood CO2 goes down, okay, down. So which way are we going to drive the equation? This went down. We need it to go back up, so the equation's going to go to the left. So all of this free hydrogen ion that was nicely floating around the blood, now we've got to make it carbonic acid so that we can go make more CO2. Now, as we do that, we're going to reduce the amount of hydrogen ions that was in the blood because we had to use them to do this and make more CO2. So the blood hydrogen ion concentration decreases, which causes the pH to go up. It becomes more alkaline. Does that make sense? The cheat sheet version is when this goes down, this goes down. So that's a lot faster and a lot easier. But I wanted to at least teach you why that happens. Okay. Okay, let's look at it from the reverse. Decreasing respiratory rate. So let's say we slow down our breathing, maybe to the point of we're not breathing rapidly enough or deeply enough. So that results in an increased amount of CO2 being retained in the blood because we have to exhale regularly to get rid of that CO2. If we're breathing too slowly or too shallow, then we don't get proper exhalation of the CO2 and it starts to build up in the blood. So if this goes up, we're going to drive the equation to the right because we need to kind of balance it out and make this go up. Cheat sheet version, CO2 goes up, hydrogen ion goes up. Anytime the hydrogen ion concentration goes up, the pH goes down. So it drives the equation to the right, the blood concentration of hydrogen ion goes up, and the pH goes down. So make sure you can kind of follow all of that from, we've got this specific change in the respiratory rate that's going to have this effect on carbon dioxide in the blood, which is going to have this effect on hydrogen ion concentration in the blood, which is going to lead to this change in pH. You should be able to do those steps, okay? And here it all is in a nice, lovely picture format. So this one talks about an abnormal increase in respiratory rate. So she's breathing rapidly or she's breathing too deeply. So she's getting rid of too much CO2. We have more CO2 being expired, which means less CO2 in the blood. Okay, so carbon dioxide went down, so the whole equation went to the left. Chemical equation driven to the left. You can see we're not looking at double-headed arrows anymore. Now everything's moving to the left because this reactant over here went down. So now everything's got to move that way to bring it back up. So we get a reduction in hydrogen ion concentration, which leads to an increase in pH. Okay, opposite scenario. Abnormal decrease in respiratory rate. She is now breathing too slowly or too shallow, so she doesn't get a lot of CO2 being exhaled, which means it increases in the blood. If we're not breathing it out, it's going to stay in the blood. So CO2 goes up, chemical reaction is driven to the right, which means hydrogen ion concentration also went up. Increased hydrogen ion concentration means a decrease in pH. Okay? This one over here, increase in respiratory rate is going to lead to alkaline conditions, okay, because the pH goes up. Decrease in respiratory rate is going to result in acidic conditions because the pH is going down. Okay, so chemical buffers. Okay, so we talked about regulating the pH through the kidneys for our fixed acids, right? Or regulating the pH through respiration for our volatile acids. That's called physiologic buffering. Physiological buffering systems is when we use those systems, like the urinary system with the kidney or the respiratory system with the lungs, to help kind of buffer or regulate the pH of the blood. The physiologic processes in the kidneys take hours to days to happen. It's not a quick fix. Those in the respiratory system, that can change fairly quickly. It takes like on the order of minutes to respond. So while we wait for those physiological systems to kind of kick into gear and do what they need to do, we have chemical buffering systems. Chemical buffering systems act quickly and temporarily, and they're not to be used for long term at all, to prevent pH changes that would occur in response to the addition of an acid or base after a meal. So we use our chemical buffering kind of in the meantime, kind of on a short-term basis before our long-term physiological systems, being the respiratory system, the urinary system, really kind of kick into gear and take over. Okay, so molecules that make up the chemical buffering systems are composed of a weak base and a weak acid. A weak base is something that can bind excess hydrogen ions. And a weak acid is one that can release excess hydrogen ions. So you'll see these a lot, like in intro chemistry, or maybe even the beginning of 2401, talked about a proton donor and proton acceptor, where the proton donor is the acid and the proton acceptor is the base. That's what we're talking about here. Proton acceptor, with a proton being a hydrogen ion. The weak base can take on a hydrogen ion. The weak acid can donate or release one. Okay, chemical buffers cause both a temporary and limited adjustments until the body can eliminate the excess acid or base through the physiological buffering system. So these guys kick into gear first, while those physiological systems kind of get ramped up and going until they're fully effective. There's three main types of chemical buffering systems. You've got the protein buffering system, the phosphate buffering system, that's primarily within our cells, and then the bicarbonate buffering system and the extracellular fluid, primarily in the blood. So let's look at the protein buffering system. This is part of the chemical buffering system. It's made up of proteins. This can happen either in our cells or our blood plasma, because that's the two places where we had proteins. So this shows you the general structure of a protein. You've got an amine group that's got nitrogen and hydrogens. So you got a central carbon. This R just means it's something else. Doesn't even matter what it is. It's something else. And then you've got the carboxyl group, which is a carbon with a double bond to oxygen and the OH hydroxyl group. So this is a very basic structure of what a protein looks like. It's important for the buffering system because the amine group, and sometimes you'll see this called amino group, this part with the NH2, this acts as a weak base. So this NH2, this nitrogen right here, it can actually accept a third hydrogen ion if it needed to. It's cool if it doesn't. It's perfectly fine like this. But if there's a lot of free hydrogen ions floating around, it can totally take another one to make this NH3. If you look at the other end of the protein, the carboxyl group, or sometimes you'll see it called the carboxylic acid, this acts as a weak acid. So this hydrogen that's on this OH group, this is free to pop off and donate a hydrogen ion. Now again, it's totally cool if it doesn't. It can stay just like this. But if you're in alkaline conditions and you really need hydrogen ions, this part of the molecule right here is more than willing to give up this hydrogen ion. Okay, so this is one molecule. It's one protein. But on one side of it, it's a weak base, meaning it can take on an extra hydrogen. And on the other side, it's a weak acid, meaning it's willing to donate a hydrogen ion. So let's look at that in conditions of changes in acidity. Okay, so let's say everything's going along and then bam, things become acidic. We get this huge contribution or a large addition of hydrogen ions. If it gets acidic, the amino group, the weak base, is going to act as a proton or a hydrogen ion acceptor. So this part of the protein can accept a hydrogen and become NH3. So we would just tack this hydrogen ion right on there. Now what that does is it reduces the amount of free-floating hydrogen ions in the solution, and that's going to increase the pH. It's going to make it less acidic because it only affects the pH when it's a free-floating hydrogen ion. Once it attaches to something, it's not contributing to the pH anymore. So then we'll look at the carboxyl group. If you get into a situation that's very alkaline, now we need more hydrogen ions. The carboxyl group is willing to donate this hydrogen ion right here. It can pop off, so again, it's this right here. It can pop off and it will join this, which would give you H2O. So this would look like C double bonded to O, bonded to this, it would have a minus, COO minus, and then additional water. And that's how it can neutralize and bring this back towards a more neutral or acidic pH if we're in alkaline conditions. So this is just kind of a refresher it kind of explains the difference between a strong acid versus a weak acid and a strong base versus a weak base. So I'm not going to read through it, but it's here if you need it, because I know acids and bases are something that we kind of covered, but it was a while back, and so in case you kind of need a refresher on things, you can read through this. So moving on to the next chemical buffering system is the phosphate buffering system. The phosphate buffering system is found primarily in the intracellular fluid, so found within the cells. It's especially effective in buffering metabolic acid produced by the cells because that phosphate group, the PO43-, is the most common anions that you have in the cells. Phosphate buffering system is, just like the rest of them, made up of a weak base and a weak acid. So hydrogen phosphate is H2PO4-. This is a weak base. It can accept an additional hydrogen ion. It's got one, but it can accept a second one. Dihydrogen phosphate, H2PO4, is a weak acid because it already has the second hydrogen ion, and it's willing to give up one of them should it need to. Okay, so we've got hydrogen phosphate that can accept another hydrogen, so it acts as a weak base. Dihydrogen phosphate that can donate a hydrogen ion, so it acts as a weak acid. So if the conditions present themselves where they're strongly acidic, the hydrogen phosphate can come in and it can take this hydrogen ion and attach it to itself. So the HPO4 becomes H2PO4. So it's removed that hydrogen ions from the solution, which reduces its acidity. Okay, so the weak base took on some of the hydrogen ions and kind of neutralized it. So this is its job as a buffer. Okay, if instead you get an alkaline condition, so you get a strong base there, then the dihydrogen phosphate, H2PO4, is willing to donate one of these hydrogens, so it pops off and it comes over here, and that gives you H2O. So when this hydrogen, one of them, jumps over here, it becomes H2O, and then it leaves you with just hydrogen phosphate. So it's one of these two molecules. This is the weak base, this is the weak acid, and just depending on the circumstances you're in, if you're in acidic conditions or if you're in basic conditions, those molecules can kind of go into action to either accept a proton or donate a proton. And again, when I say proton, I mean hydrogen ion. And that works to just kind of keep the pH where it needs to be so that you can add acid to something without it drastically changing the pH of it, because you have buffers around. So let's say you pour in a bunch of acid, you have buffers around that consist of a weak base, and they can soak up some of those hydrogen ions so it doesn't instantly become and stay acidic. Now, if you poured that acid into a solution that didn't have a buffer, then definitely the pH is going to go down and it's going to be very acidic. So buffers allow for the addition or the removal of hydrogen ions in a way that it doesn't change the pH, because we can't afford to change the pH very much within our cells and within our blood. And then finally, the bicarbonate buffering system. This one is found primarily in the extracellular fluid. This one you have bicarbonate or HCO3-, so we've seen this before. And then this acts as the weak base because it can take on an additional hydrogen ion. And so its kind of companion over here is carbonic acid or H2CO3. So H2CO3 has two hydrogens, so this acts as the weak acid because it can take one of these and donate it if it needs to. So if somebody adds a bunch of acid to the environment, here's the weak base, here's bicarbonate, it can tack on this hydrogen to itself to become H2CO3, neutralizing that environment. If it becomes pretty alkaline, you've got carbonic acid that has H2CO3, it can donate one of these hydrogen ions over here to form bicarbonate and water, and again, neutralizing it. Okay, so they all work the same way. Each one of the chemical buffering systems is made up of a weak base and a weak acid, where the weak base can take on a hydrogen to neutralize it, the weak acid can donate a hydrogen to neutralize it. It's just a matter of, so know the names of the three types, know where you find them predominantly in the extracellular versus the intracellular or what have you. So their name, where you find them, and then the molecules involved. So like for the protein, it was just proteins. That's all that was involved. But you had the amino end and the carboxylic acid group. So you got two different sides of the protein. In this case, the two different molecules are going to be bicarbonate and carbonic acid. And the other one, we talked about hydrogen phosphate versus dihydrogen phosphate. So know the molecules involved, which one's an acid, which one's a base, and kind of what their role is. Okay, a little note down here about chemical buffering. The modulation of the pH changes by the chemical buffers usually allows time for the kidneys to alter the excretion of the hydrogen bicarbonate, okay, or for the respiratory system to adjust the expiration of CO2. So they are there just to basically buy ourselves some time for those physiological systems to kick in, the kidneys and the respiratory system. All the chemical buffering systems are limited in the amount of acid or base that they can buffer. If the buffering capacity is exceeded, then the pH levels may continue to change or increase or decrease beyond the normal limits. Those changes in pH are referred to as acid-base disturbances. So in a perfect world, we get, changes in the acid composition, let's say, but our chemical buffering systems kick in right away, and it buys us just enough time for our physiological buffering systems to kick in, and everything transitions very smoothly. If we use up all of the capacity of our chemical buffering systems before our physiological buffering systems have come into play, that's when you get that acid-base disturbance. Okay, so an acid-base disturbance occurs whenever the buffering capacity of chemical buffering system is exceeded, and we haven't yet kicked in with those physiological buffering systems. So you get a temporary change in the blood hydrogen ion concentration that results in a change in the blood pH beyond its normal range. Its normal range is 7.35 to 7.45. So if there's a change and our chemical buffering systems are exceeded and our physiological buffering systems have not yet kicked in, and we get a change either above 7.45 or below 7.35, that's an acid-base disturbance. Okay, it's specifically referred to as acidosis if you have an arterial blood pH below 7.35. Because remember, lower levels are acidic, higher levels are alkaline. So acidosis is blood pH below 7.35. Alkalosis is blood pH above 7.45. Okay, in response to a temporary acid-base disturbance, the physiological buffering systems of the kidneys, the respiratory, or both have to operate to offset that disturbance. So eventually these physiological systems are going to kick in and kind of do their job to buffer the pH of the blood. Okay, so when the physiological buffering systems actually do their job and they return the pH back to its normal level, that's called compensation. Okay, so ideally we don't want it to change. Ideally, you give, let's say, an increase in acid input, you have your chemical buffering systems kick in, and before they are, maxed out, your physiological buffering systems kick into play, and your pH never leaves that range of 7.35 to 7.45. Okay. If we max out our chemical buffering systems before the physiological buffering systems kick in, then you might get a shift either above or below that range. That's called an acid-base disturbance, acidosis if it goes below the normal range, alkalosis if it goes above the normal range. But when our physiological buffering systems kick in and they get it right back down into that normal range, that's called compensation, okay? If the physiological buffering systems are not effective in returning the pH to normal, then that disturbance is referred to as being uncompensated. If our kidneys and our respiratory system don't get the job done and our pH stays outside of that normal range, it's referred to as uncompensated. When that happens and you have this persistent change in pH, it's called an acid-base imbalance. So if you get an acid-base disturbance, that doesn't get compensated, it doesn't get corrected, then now it's called an acid-base imbalance. An acid-base imbalance that goes for any extended period of time can be life-threatening. So if the pH reaches values either below 7 or above 7.7, it can be fatal within hours. We do not have a lot of leeway when it comes to the pH of our blood. There's 4 types of acid-base disturbances. 2 of them are like acidic, and 2 of them are associated with being too alkaline. And then 2 of them are associated with respiration, and then 2 of them are associated with metabolism. So we have respiratory acidosis, respiratory alkalosis, metabolic acidosis, and metabolic alkalosis. So we'll start with respiratory acidosis. This is the most common of the acid-base disturbances that you'll find. This one happens because of impaired elimination of carbon dioxide by the respiratory system. Okay, so respiratory acidosis happens whenever CO2 in the arterial blood goes up. So it would have to be above about 45 millimeters of mercury. So normal is going to be between 35 and 45. So anytime it goes above that 45, that's considered kind of too elevated. Possible causes are injury to the respiratory center in the brain if you're just not getting those nervous signals to breathe at the appropriate rate or depth. Injury or weakness to the muscles involved in breathing, which happens in muscular dystrophy, or late stage ALS, because that diaphragm is skeletal muscle. And in ALS, you're losing the nervous input that communicates with the skeletal muscle. So basically, there's no nervous input telling, or it fades away. So you'll have less and less nervous input telling that diaphragm to contract and relax. So you're just not getting communication with the muscles involved in breathing. Airway obstruction, and then emphysema. So all of those can affect the rate and the depth of your breathing. And if you're not doing it, if you're not breathing sufficiently, then you're not going to exhale sufficient levels of carbon dioxide. So what happens is, if you're not getting it out, the CO2 then accumulates in the blood. Okay? Carbon dioxide accumulates in the blood, and it ultimately causes an increase in carbonic acid, which leads to a subsequent increase in hydrogen ion concentration. So if we are breathing too slowly or too shallow, we're not eliminating enough CO2. So this increases in the blood. And remember from my cheat sheet, I said this goes up, that means this goes up. And anytime you see an increase in hydrogen ion concentration, that means your pH goes down, which is associated with acidosis. So it's acidosis because we're lowering the pH. It's respiratory because that's what started it, was insufficient respiratory rate or depth. The next one is respiratory alkalosis. So respiratory alkalosis is when the level of CO2 decreases in the blood. So it's going to go below that 35 millimeters of mercury. And that's going to happen whenever there's an increase in respiratory rate or depth. So usually that's hyperventilation. So severe anxiety, aspirin overdoses, because aspirin stimulates certain areas in the brain that stimulate your respiratory center. So those are going to be the 2 main causes of hyperventilation. But basically, you are going to exhale too much carbon dioxide. So you are just breathing way above and beyond what your body actually demands, and you get rid of too much CO2, which means the CO2 in the blood goes down. Okay, so if this goes down, then your hydrogen ion concentration goes down, and that means your pH goes up, and you become alkaline. Okay, so respiratory alkalosis. The next one is metabolic acidosis. Metabolic acidosis is the most common metabolic acid-base disturbance that is going to happen as a result of a decrease in bicarbonate. Okay, so this can result from excessive loss of bicarbonate, but more often than not, it occurs when there's an accumulation of fixed acid. So it may have to do with the fact that maybe your body lost too much bicarbonate, but mostly it's going to be if you just accumulated too much hydrogen ions. So the excess hydrogen ion binds with the bicarbonate that you already had to form carbonic acid. So that causes a reduction in their bicarbonate levels in the blood. So metabolic acidosis happens when the arterial blood levels of bicarbonate fall below 22 milliequivalents per liter. So your normal is going to be 22 to 26. If you take in too much acid, that acid's going to bind this up again to form carbonic acid, which is going to leave you with insufficient amount. So when it goes below 22, then it's considered metabolic acidosis. Okay, possible causes. There's increased production of metabolic acids through diabetes. So if you have diabetes, then you may have all the blood sugar in the world, but your cells don't see it. Either the insulin didn't come out, or for whatever reason, your cells just don't realize the glucose is there. So even if your blood sugar's through the roof, from the perspective of your cells, there's no glucose because they don't have it to use. So their only other option then is to start breaking down fatty acids. And when they start to break down all the fatty acids, it produces those keto acids. So those keto acids are one of the metabolic waste products. And you get a buildup of keto acids, and then you start, that's where you pick up right here. All the excess acid is going to bind to bicarbonate. Anaerobic respiration, that's the one that produces lactic acid. Excessive alcohol consumption, decreased elimination of acids due to renal dysfunction. If your kidneys aren't working right, you can't get rid of the acid, it's going to accumulate in the blood. Or increased elimination of bicarbonate as a result of severe diarrhea. So any of those are possible causes, and it's going to lead to metabolic acidosis. This explains a little bit about lactic acidosis and ketoacidosis. I've already kind of explained these earlier in the lecture, but I do want you to read through so you kind of get all of this information. Keep in mind that ketoacidosis, I think there's newer derivatives of dietary fads these days that do it, but Atkins, or if you try to go completely carb-free, it basically looks like diabetes to your body. If you're not providing the carbohydrates that it needs, it's forced then to start metabolizing fatty acids, which I think is the big draw on the appeal. People want their fat to be metabolized, but one of the byproducts are keto acids. And you can tell after this lecture that keto acids can lead to ketoacidosis, and it can affect your pH of your blood. So anyways, just kind of read through this. Again, nothing of this is completely new because I've already kind of touched on this as we've gone through, but about, lactic acid and keto acids. The last one is metabolic alkalosis. This is defined as arterial blood bicarbonate levels that exceed 26 milliequivalents per liter. So again, it was from 22 to 26. As you go above 26, you're in metabolic alkalosis. This is going to be caused by a loss of hydrogen or an increase in bicarbonate through vomiting. That's going to be the most common cause. Increased loss of acids by the kidneys if you are overusing diuretics, or increased alkaline input from consuming a large amount of antacid tablets. So any of those can lead to metabolic alkalosis.